Greetings everyone and welcome to another exciting genetic engineering and biotechnology news webinar. Our presentation today is entitled CRISPR-Cas9 Cell Therapy, the Next Generation of Cures. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. The use of genome editing technologies has exploded in the past several years, due in large part to the discovery of the CRISPR-Cas9 system. This DNA editing technology has allowed researchers to rapidly and efficiently introduce changes into the genomic sequence of numerous organisms, highlighting the immense therapeutic potential for treating various disease states. Yet with most technologies that are applied to cells exogenously, researchers often struggle with finding appropriate non-viral techniques to get the molecules past membrane barriers and deliver to the appropriate cellular compartment. Let's meet our panel for today's webinar, who will describe current research methodologies that are looking to translate recent successes with the CRISPR-Cas9 system into disease therapeutics. Additionally, we will hear how nucleofaction technology can improve the delivery of gene-editing DNA while increasing transfection efficiency and allowing researchers to use the most relevant cell lines for their works. Our first speaker today is Matt Porteous, Associate Professor of Pediatrics at Stanford University. Dr. Porteous will provide us with an introduction into CRISPR mechanism and the approaches investigators are utilizing to facilitate the genome editing technique into clinical therapies to treat an array of diseases. Our second speaker will be Gregory Alberts, Global Subject Matter Expert for Transfection and Genome Editing at Lonza Bioscience Solutions. Dr. Alberts will describe the advantages of nucleofaction transfection technology and how it can improve the laboratory workflow for gene editing researchers. Before our speakers get started, I want to encourage you to submit questions for our Q&A session at the end of the presentations. We'll try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All right, without any further ado, let's get our webinar underway. Matt, take it away.
Good day. My name is Matthew Porteous. I'm an Associate Professor of Pediatrics at Stanford University, and I'm excited today to talk to you about CRISPR-Cas9 Cell Therapeutics, the next generation of cures. Now, while today I will be talking about my work in my academic lab at Stanford, I've been fortunate enough to become involved in a biotech company called CRISPR Therapeutics, and according to Stanford University policy, I have to disclose that potential conflict of interest, and so that's what I've done in this slide. So today I want to talk to you about genome editing, which is a way of precisely changing the DNA sequence of a cell. And what I want to point out in this first slide is that, in fact, from the cell's perspective, we're mutagenizing the genome. We're changing the sequence of the DNA from what it was into something that we want. And the fundamental process works in the following way, which is that you design a specific protein or nuclease that will recognize a target site in the genome and create a double-stranded break at that site. The cell then recognizes that double-stranded break and repairs it in one of two primary ways. The first is by non-homologous end joining, shown on the left part of the slide. And through the iterative and non-homologous end joining works by essentially stitching the two ends back together. This is usually an accurate form of repair, but if it's accurately repaired, it means it reconstitutes the binding site and the nuclease can come back and create another double-stranded break. And so what you end up with is an iterative cycle of break and repair that generates small insertions and deletions at the site of the break. And this is a way of inactivating specific genetic elements. The other way that the break can be repaired is by homologous recombination. And in this mechanism of repair, if you provide a donor sequence, the cell will use that donor sequence through what is conceptually a copy and paste mechanism and make a copy of the donor DNA and insert that copy into the genome, repairing the break. And by designing the donor DNA with small nucleotide changes, including single base pair changes or large cassettes of genes, one can create precise nucleotide changes in the genome. And the break stimulates this process by over 100,000-fold. So what's interesting, though, is that while we're using a DNA double-stranded break to initiate genome editing for our research and therapeutic purposes, nature also uses double-stranded breaks for this purpose. These are some examples of how double-stranded breaks are used by nature are shown in this table. Probably the most interesting one is that the RAG1, RAG2 complex will create a double-stranded break to stimulate VDJ recombination. But interestingly, in mammals, those breaks are repaired by non-homologous end joining, whereas in other species, including birds and sharks, the breaks are repaired by homologous recombination. I think it's possible that by studying these natural mechanisms of genome editing that we'll be able to improve not only the efficiency of our research in therapeutic editing, but perhaps even the specificity as well. So I'm a pediatric hematologist oncologist, and I've been focused on trying to develop genome editing as a potential way of curing genetic diseases. So monogenic diseases are diseases caused by mutations in single genes. They permeate all of medicine. There's estimated that there may be actually up to 10,000 such diseases, and they span all of medicine. And the exciting thing about genome editing is because it allows us to change the DNA sequence in a precise fashion, it gives us a way, perhaps, to use genome editing to correct disease-causing variants and thereby cure patients of their disease. So there's some simple principles that we use when we're trying to optimize the frequency of genome editing, and those are shown in this slide. In the top part of the slide, the first principle is that to get high frequencies of genome editing, whether it's by non-homologous end joining or by homologous recombination, you first need to introduce sufficient nuclease into the cell. If you don't create breaks, you don't get editing. And so what we show here is that as you decrease the amount of nuclease in the cell, your overall editing frequency goes down. So if you encounter a situation in which the editing frequency is low, it's probably because either your nuclease is inactive or your nuclease is not in the cell at sufficient quantities to find the target site. One of the simplest ways to balance the ratio of homologous recombination versus NHEJ-mediated editing is to introduce sufficient donor into the cell. So by giving the cell lots of donor to work from, the cell will use that to repair the break, and actually you can get frequencies of homologous recombination-mediated editing that exceed the frequency of NHEJ-mediated editing. So figuring out ways to get the donor into the cell is very important. Now there are a number of different alternative nuclease platforms that are now available, and these are schematized in this slide and summarized in this table here. And the CRISPR-Cas9 is the newest platform, and was first discovered in 2013. And while it works through a similar mechanism through all the other nuclease platforms, that is, it induces a specific double-stranded break in the cell, the reason that it has transformed the field of genome editing is that it is incredibly simple to engineer a nuclease, a CRISPR-Cas9 nuclease that binds a specific site in the genome. So the tau effector nucleases were easier than zinc finger nucleases, which were easier than homing endonucleases or megatau nucleases. In terms of activity on the on-target sites, a well-designed nuclease of all these platforms works well. And in our hands, we find that actually the specificity of the CRISPR-Cas9 nuclease is quite high and probably higher than these other platforms. And finally, in the last column, I list some companies that have purchased or acquired IP around the different nuclease platforms as this hopefully moves into the commercial realm. So before describing how we use the CRISPR-Cas9 platform, I want to quickly describe where this system comes from. And it's an amazing story that actually bacteria, just like we have, we have an adaptive immune system. But in the bacteria, the way the adaptive immune system works is that when an incoming foreign pathogen invades the bacteria has a system to cleave the incoming nucleic acid into small pieces, and those small pieces get integrated into the bacterial genome, which then get processed by the CRISPR pathway into small, what we call cRNAs. These cRNAs hybridize with what's called a tracer RNA, and this 2 RNA complex then complexes with a Cas9 protein, and the next time the invading nucleic acid comes into the cell, this CRISPR-Cas9 complex is poised to cleave that DNA into small pieces. And amazingly, this system adapts incredibly well into essentially every cell type that's been tested. So, like I said, the way we use the CRISPR-Cas9 system is actually shown on the right, in which we fuse the CRRNA and the tracer RNA into a single RNA, so a short guide RNA. And then this approximately 199 nucleotide RNA molecule complexes with the Cas9 protein to form the CRISPR-Cas9 complex. There's some features to the CRISPR-Cas9 system, and I will focus here on the system that's derived from the bacteria Strep pyogenes. So usually the guide sequence is 20 nucleotides long. This is the sequence of the RNA molecule that determines where in the genome the nuclease will make a break. But this system can tolerate 3 to 4 mismatches, and sometimes perhaps even more. And sometimes the guide RNA can actually be shortened down to 19, 18, or 17 base pairs. And interestingly, by shortening the guide RNA, you increase the specificity. Now, sometimes when you shorten the guide RNA, you reduce the on-target activity. So this is something that has to be empirically tested. The nuclease activity, the activity that actually creates the double-stranded break, lies within the Cas9 protein itself. It leaves a blunt end in the DNA, and this is slightly different than the other nuclease variants. And there are multiple new Cas9 variants being discovered continuously. These include Cas9 variants, natural Cas9 variants from other species, and now increasingly scientists using protein engineering to design novel, non-natural Cas9 variants that have different properties that may be useful to us in the future. So when we started trying, the CRISPR-Cas9 system has been incredibly useful in cancer cell lines. But as we started trying to use the system that was described in cancer cell lines, we found that it was absolutely inactive. And so our hypothesis was that that we were not able to deliver enough of the active nuclease into our primary T cells and hematopoietic stem and progenitor cells as DNA plasmids. And so in a collaboration with Agilent Technologies, who had the ability to synthesize the 99 nucleotide guide RNA, we explored whether direct delivery of the RNAs rather than as DNA plasmids would solve the problem of using the CRISPR-Cas9 system in hematopoietic stem and progenitor cells and T cells. And in this slide, what we show first is in the cancer cell line that when we use guide RNAs that have been modified to resist exonuclease degradation by the intracellular exonucleases, we get significantly higher frequencies of insertions and deletions for the same amount of guide RNA. So across 3 different genes, we see the same phenomenon, where the unmodified guide RNA gives very low activity, and the stabilized guide RNAs, the MS and MSP, give very high frequencies of genome editing. So this was in a cancer cell line, which we got high frequencies of editing using plasmids. But when we went into CD34 positive human hematopoietic stem and progenitor cells, we can see that using the chemically modified synthesized short guide RNAs was highly effective. So across 2 different genes, what we show is that when we deliver the CRISPR system as plasmids, we get no activity, but when we deliver the CRISPR system with the Cas9 delivered as an mRNA and the guide RNA delivered as a stabilized molecule, either the MS or MSP variant, we got higher frequencies of insertions and deletions. When we then looked in T cells, so one potential application of this strategy is to inactivate genes that might be therapeutic. And so there's been several publications, and in fact a clinical trial using zinc finger nucleases showing that if you inactivate the CCR5 gene, which is a co-receptor for HIV, you might be able to engineer resistance to to infection by HIV. So we, and this was done with zinc finger nucleases, so we looked at whether this could be done using the CRISPR-Cas9 system. And what we found is that if you made two guide RNAs that both targeted the same CCR5 gene but about 200 base pairs apart, that in T cells, we could get over 90% of the alleles modified by these two guide RNAs. Most of the modifications were deletions between the two breaks, but we also saw all combinations of modifications, including inversions in which the fragment dropped out and inserted itself back into the locus in an inverted location. But nonetheless, by using two guide RNAs, using this Cas9, CRISPR-Cas9 system with the synthetic guide RNAs, we were able to get 90% allele knockout in T cells. So we're interested in using homologous recombination rather than just simply breaking genes. And what I show in this slide is that there's more than one way to think about homologous recombination. And over the next few slides, I'll show you examples of each. So one can use homologous recombination to fix a point mutation, so just make a small number of nucleotide changes. One can use homologous recombination to target the cDNA for the gene back into the locus. So you knock in the wild-type cDNA into the mutated gene, thereby correcting it. And one could use the concept of a safe harbor, where you take an entire expression cassette and knock that expression cassette into some specific location in the genome so you don't worry about it going into other sites. And again, I'll give examples of each of those three. So I mentioned earlier that we need to deliver the donor vector. And in the cell types we work with, delivering the donor as naked DNA causes toxicity. So we had to figure out a way of getting the donor for the homologous recombination into the cells. And what we've adopted is the use of the adeno-associated virus. So the system works by delivering the CRISPR-Cas9, and I'm going to show you data why we do it as a ribonucleoprotein complex. So the Cas9 as a protein, the guide RNA as an RNA molecule, which we introduce by nucleofection using the Lonza device. And then we deliver the donor using a recombinant AAV virus. So first we showed that AAV6, one of the serotypes of AAV, is able to transduce, is able to get into CD34 positive cells from peripheral blood, bone marrow, and cord blood at high frequency. So in a way that was non-toxic and did not activate the cell's sort of innate immune system. So then we looked at whether we could use the CRISPR RNP system combined with AAV delivery to knock in a cassette into a specific locus. And to do this, we made a AAV which had a strong promoter driving GFP, but one could think about GFP being any therapeutic transgene. And what we found is that using the combination of AAV and the CRISPR system at the HBB locus, we could get on average between 15% and 30% targeted integration of the cassette into the locus. What we found is actually using delivering the CRISPR-Cas9 as an RNP rather than as an all-RNA system, we got about 50% higher frequencies of editing. And across 5 different genes, on average, we get about 17% targeted integration, although next year this number will probably be higher as we get better and better at the process. What's interesting about using the RNP versus the all-RNA system is you also improve specificity. By delivering the CRISPR system as a protein complex, the half-life in the cell is much shorter than if it's delivered as an mRNA, which reduces the probability that it causes breaks at off-target sites. And so we see improved on-target activity, and we see improved specificity. So now I want to show you a couple slides about how we can use the process to change specific nucleotides in the genome. And for this, we have Cas9 disease called sickle cell anemia. Sickle cell anemia is an autosomal recessive disease in which every patient has an A to T change at codon 6 of the HBB gene. We designed guide RNAs to recognize the site adjacent to the sickle cell variant, and we made a AAV vector that fixed the variant, introduced changes to prevent the nuclease from recutting the gene after we fixed it. This is a very important process, a very important feature of the donor molecule. And then we introduced our HBB-specific CRISPR-Cas9 along with our AAV vector into stem cells from sickle cell patients, and we found the following. On a population level, only 10% of the alleles remained unchanged. About 40% of the alleles developed an insertion and deletion, so we broke the gene, but about 50% of the alleles were corrected. We were able to change the sickle variant back to the wild type variant. What's important about this frequency is it's estimated that 10 to 20% correction in stem cells should cure the disease. And they showed importantly that these modified cells are able to engraft into mice, showing they retain their hematopoietic stem cell potential. And so our modification frequency of the population of cells was 44%, and after transplantation into mice, we found that the corrected allele frequency was approximately 40%. So we did not lose the corrected cells after engraftment. So a second approach I want to describe is the idea of knocking in a cDNA to functionally correct a gene. Now, why would you want to do this? Well, SCIDX1, which is a disease called severe combined immunodeficiency, or sometimes known as bubble boy disease, because David Vetter, who's our most famous patient with bubble boy disease, had to live in an isolated bubble to prevent him getting an infection, and he lived in Houston, so NASA actually even built him a spacesuit so he could walk around but still be isolated in the environment. Now, in contrast, sickle cell disease, mutations that cause SCIDX1 are scattered throughout the gene. That is, every patient basically has a unique mutation. And so designing a specific correction system would be very challenging. So we and others adopted a hybrid strategy in which a hybrid genome editing gene therapy strategy in which we take the wild type cDNA and knock it back into the endogenous locus. So the gene will be expressed from all of its natural regulatory elements. And looking at the SCIDX1 gene, this strategy should fix over 97% of the mutations that cause the disease. So we made some different donor vectors that either had the codon-optimized cDNA alone or had selectable markers. And then using quantitative molecular techniques, what we showed with the simplest donor is that we were getting, on average, across here, we showed 5 different sources of CD34 cells, an average of 33% targeted integration of our cDNA into the locus, and that this targeting frequency stays stable over time. And thus, we think, in data I'm not going to show, we show that by knocking in the cDNA into the locus, it actually restores full functional activity to the gene. So one of the important things that we're going to have to do as we take what we think is very promising preclinical data to the clinic is we have to be able to scale up to clinically relevant cell numbers. So in all of our experiments, we've been using the Lonza 4D device, which allows you to manipulate between 100,000, or if you want to really scale up, 20 million cells at once in volumes of 20 to 100 microliters. We were fortunate to collaborate with the Lonza scientists to test their new large volume device, which now can modify between 20 million and up to 2 billion cells at once in a larger volume of 1 to 20 mils. And what we found is that the viability of the cells after electroporation was equivalent between the two devices. The indel frequency was equivalent between the two devices in T cells and CD34 cells, and the homologous recombination frequency, the frequency of genome editing by homologous recombination using the two devices were equivalent. So what the LV device allows us to do is to establish our parameters and our conditions to optimize the frequency of editing using the small scale device and then easily translate that up to the big scale device. We had to make no changes in programs, conditions, or any other variables to do this. To end, I just want to contrast the issue of ex vivo versus in vivo editing. We have focused on ex vivo editing, whereby one would remove your cell type of interest, modify it outside the body, and transplant that into the patient. And the advantages of this is that it's easier to control delivery, it's easier to measure on-target efficacy, and easier to measure off-target effects. The challenge is really, is the organ or the disease that you want to cure amenable to cells that you know how to bring outside the body and transplant back in? And there are a number of different cell types for which this is possible, including hematopoietic stem cells, keratinocytes, mesenchymal stromal cells, and so on. But there's a number of other cell types for which this is not possible. For that, one would have to go to an in vivo approach. And the idea here is to deliver the genome editing machinery directly to the patient to modify cells in situ. And this might be necessary for cells that we do not know how to remove from the body and transplant back. For instance, cells of the eye, brain, and liver. It also has the advantage that, of course, you just have to manufacture your vectors to deliver into the body. You don't have to manufacture your cells. The challenges with in vivo editing is you have a lower ability to monitor efficacy and off-target effects. You have to figure out how to deliver your genome editing components to the relevant cell types in the body, and you have to worry now more about nuclease and vector immunogenicity, because now the patient's own immune system might recognize the genome editing components as being foreign. One of the things that is always discussed, and I think appropriately, is the issue of if we're creating a double-stranded break at our on-target site, is it possible we're creating double-stranded breaks at off-target sites that might be problematic? And what I think is a very important paper, which is going to be published in Molecular Therapy Methods in Clinical Development by my colleague David DeGiusto here at Stanford, but this work was done with John Zaya and collaborators at Sangamo Biosciences down at City of Hope. And this was a protocol they developed to use ZFMs to target CCR5. I mentioned them earlier in my talk, to create an immune system that's resistant to HIV. And what they found is that their ZFMs created insertions and deletions at several other sites in the genome. But what they showed through a combination of bioinformatics, which is that these off-target sites seem to have no biologic importance, combined with functional studies showing that populations of cells that had undergone modification with these ZFNs were completely equivalent to unmodified cells, the FDA said that they would give approval to a phase 1, phase 2 trial using these ZFNs. And I think that is our first evidence of what our standard is for specificity in terms of bringing a nuclease-based genome editing approach to the clinic. The thing I want to point out is that while we have to be concerned about nuclease off-target effects, we also have to put the mutagenic effects of the nuclease into context of what goes on in our bodies. So first of all, we are all born with over 2 million individual nucleotide variants. So we are very diverse to begin with variants all over our cells. We have 40 trillion cells in our body at any one time, and through the natural actions of life, including breathing oxygen and burning energy, we generate about 1 mutation per cell per day. That means we're generating about 40 trillion mutations in our body any given day. If we think about transplanting our cell, doing a hematopoietic stem cell transplant, we have to give what we call conditioning chemotherapy to eliminate the host hematopoietic stem cells. And I calculate that this may actually generate around 1,600 trillion new mutations in the body in order to create space. So if you contrast that, we might be generating on the order of 10 million de novo mutations using our nuclease. This, of course, pales in comparison to the number of mutations that are occurring both naturally and through our own therapeutic interventions. Now, of course, what we have to make sure of is that our nucleases aren't hitting a specific site in the genome that might be problematic. For example, a p53 gene or an oncogene or a tumor suppressor. So while we need to be concerned, I think we need also to put this in context. So as we think about developing genome editing as a therapy, we want to think about we really need to have deep understanding of the disease that we're trying to address. And once we understand the disease we want to address, we can then design a strategy that is based on, do we need to get homologous recombination to occur, or can we design a strategy that's based on non-homologous end joining mediated editing? Are we going to simply correct a gene defect, or are we going to create a new genome and give that cell new properties, something that is akin to synthetic biology? Can you do the process through an ex vivo approach, or is it going to be better to do it through an in vivo approach? You need to understand what cell type needs to be modified. For blood diseases, we need to modify the hematopoietic stem cell. But interestingly, we know from certain neurologic diseases, neurodegenerative diseases, that actually modifying the hematopoietic stem cell can be effective. So understanding what cell type can deliver a therapeutic effect is important in understanding what cell type need to be modified. And then we need to know what frequency needs to be modified. In SCID, actually, we have experiments of nature that tells us perhaps as low as a single cell is necessary. It tells us we have a very low bar to get over. Sickle cell disease, we think we need to hit around 5 to 10%. Other diseases, we may need to be closer to 90%. So again, the disease biology is going to tell us what frequency of editing we need to achieve. And then we have to think about how you technically engineer that cell type, how you evaluate efficacy, and then how you evaluate safety and toxicity. So with that, I want to thank you for your attention. I work at Stanford University, where we are developing a center for definitive and curative medicine to bring cell-based and gene-based therapies to cure patients. We have a fantastic university, a fantastic medical school, fantastic children's hospital and adult hospital, and a wonderful environment here in Silicon Valley. And with that, I want to thank you for your attention, and I look forward to any questions you may have.
Thanks, Matt. That was an excellent presentation. I think you gave the audience some great insight into the clinical uses and therapeutic potential of genome editing systems like CRISPR-Cas9. Before we move on to Greg's presentation, I want to remind everyone once again to submit your questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. Both of our speakers will be available to answer your questions after the final presentation. All right, with that said, let's move on to our final presentation. Greg, the floor is yours.
Hi, my name is Greg Alberts. I'm the Global Subject Matter Expert for Lonza, and I'd like to continue on with our presentation to talk about CRISPR and nucleofection and the next generation model systems, the way I see things moving in the future here in the biomedical research field. So the new trends in biomedical research are really involved using primary cells in research. These are more appropriate cells to use in research. They respond more appropriately to stimuli. They express the right receptors, and so they're going to be more relevant. And then also, these results are more likely to translate into an in vivo situation because these are normal cells compared to the cell lines that people are using. So it's a way of creating a better and more accurate model system for cell-based assays. Nucleofection is a major part of this. It is an advanced form of transfection technology and electroporation. And our niche, we really excel in the transfection of primary cells. So this makes it a technology that can help you exploit these new trends. It's an excellent choice for iPSC generation, primarily because we can transfect primary cells, as well as the transfection of all the genome editing technologies such as zinc fingers, talons, and CRISPRs. So if you put all of these things together, you can use nucleofection in combination in order to generate iPSCs that can then be genetically modified, differentiated, and then used to create more complex advanced model systems. You can also generate modified cells that can be used to model and evaluate therapeutics strategies, which eventually could be used directly in cell-based therapies. So let's see how nucleofection fits into next-generation model systems. Basically, with nucleofection, you have high-efficiency transfection of primary cells and physiologically relevant cell lines. So now you can use most relevant cells for your research. You no longer have to compromise because nucleofection will help you address your transfection needs with these relevant cell types. Higher quality of cells will lead to higher quality of data and higher quality of hits. And nucleofection is superb for the transfection of all the genome editing technologies, such as zinc finger, TALENs, CRISPRs, and others. To a large extent, nucleofection is independent of what substrate you choose to use. So you can use transfect things like plasmid DNA, siRNAs, DNA oligos, mRNAs, peptides, proteins, morpholinos, QDOTs. These are all things that have been successfully transfected using nucleofection. We also have the flexibility of scale. With our Nucleotide platform, we can work from the bench top to a 96-well capacity, to a 384-well capacity, and even into our large volume, our large cell number transfection device, the LV. And so we also bring innovation into transfection. By switching to this black plastic electrode, it allowed us to make a realistic and robust 384-well transfection device. It also allowed us to do things like adherent transfection in a 24-well capacity for neurons and other cell types, and it also allowed us to create our large volume, large cell number transfection unit, the LV unit, which will allow you to transfect up to 2 billion cells straightforward and easily. With nucleofection, you can transfect virtually any cell in the body. When I put this slide together, I looked at main therapeutic areas that people were working in, such as cancers, neurology, immunology, metabolic disorders, or cardiology. I then selected representative diseases from each of these areas, and then started listing primary cell types, which we can already transfect, that might be relevant to people working in these areas. So for cancers, I chose prominent cancers like breast and prostate cancer, and we can transfect mammary or prostate epithelial cells very nicely. With neurology, we can transfect any neuron in the body, whether it's a cortical neuron like a hippocampal neuron, or peripheral neurons such as dorsal root ganglia or superior cervical ganglia. In immunology, we can transfect virtually every cell there that is relevant, such as T cells, B cells, monocytes, macrophages, dendritic cells, NK cells, and we can do this in human or mouse. So you can choose your model system, and nucleofection will be able to help you address this effectively. Things like metabolic disorders, we can transfect cells like INS1 and MIN6, which are relevant insulin-secreting cell lines, as well as other adipocytic cell lines like 3T3L1. Cardiology, we can transfect all the vascular cells like QVEX and HEMVEX, smooth muscle cells, cardiomyocytes as well. I left iPS generation and stem cell transfection for the last. Because nucleofection can transfect primary cells so effectively, we're a natural choice for iPSC generation, and that's something we do very effectively. Once you've generated your iPSCs, you then will want to be able to transfect them and manipulate them and work with them. And nucleofection is also superb at the transfection of stem cells. This slide is an example of a nucleofection in primary human T cells, cytotoxic T lymphocytes. This is an old paper. This is from 2003, and it just shows how long nucleofection has been used to work with primary human T cells. In this particular case, the researchers were studying the release of perforin into target cells, which is the means that cytotoxic T lymphocytes use to kill their target cells. So perforin is stained in green here, and using nucleofection, the researchers introduced another protein, MUNC13-4, which was fused to an RFP, a red fluorescent protein. MUNC13-4 was implicated in the release of perforin, so they were studying this. In the diagram in J, you can see that both MUNC13-4 and perforin are scattered around the cell. However, if you look at panels L and M, you can see the cytotoxic T lymphocyte in contact with the target cell. And you see this tremendous co-localization of MUNC13-4 and perforin right at the immunological synapse, allowing a direct release of perforin into the target cell, killing it. The relevant point here is that nucleofection has long been used for working with primary human T cells. In fact, that was our first kit way back in 2000. So for many years, people have been working with nucleofection and primary human T cells, and now as we move towards these advanced therapies like CAR T therapies and the need to manipulate human T cells, nucleofection is a natural. And as we create these model systems, it's also a natural for research in these fields. As I said before, nucleofection is a superb technology for the delivery of genome editing technologies such as zinc fingers, TALENs, and CRISPRs. As all these molecules work in the same sort of way. They have a DNA binding domain, and they have a nuclease activity which will sit there and deliver a nick or a double-strand cut into the DNA. As you can see here, there's different DNA binding motifs. Zinc fingers have the zinc finger DNA binding domain fused to a focal nuclease, which will allow a single nick on the DNA. You'll have two of these molecules deployed, one on either arm of the DNA. Each will deliver a nick, which will effectively generate a double-strand cut. TALENs function much the same way. They have a different DNA binding motif, but at the same time, they have a focal nuclease to generate the nick. You have one of these on either arm of the DNA to deliver a double-strand break. CRISPR is different. It has the generic Cas9 protein, which delivers a double-strand break, but it is guided to the target area by these guide RNAs, complexes with these, and then it binds the DNA and delivers that cut. No fusion protein is required here. The Cas9 is generic, and all you have to do is mix that with the appropriate guide RNA for targeting purposes. Because no fusion protein is needed, this makes CRISPR much more flexible and easily managed compared to the other technology which require the formation of these elaborate fusion proteins, actually a pair of these. So CRISPR-Cas9 is so much easier and more flexible to use, so that makes it, that is why it is sweeping the research communities as this effective technique. The relevant point here, though, is that nucleofection has long been recognized as the premier technology for delivery of zinc fingers and TALENs. In fact, if you go to the Sigma tech support and say I'm having problems with my zinc fingers or my CRISPR, the first question they're going to ask is, are you using nucleofection? Because they use nucleofection for everything. It is routine, it's robust, it is reliable, it works every time. So that's why they like this. And when you start talking about CRISPR, all the leading CRISPR researchers and pioneers, whether it's George Church or Charpentier or Doudna or Zhang Fang, they're all using nucleofection, particularly when they're transfecting difficult cell types like their stem cells, iPSCs, and things along that line. So again, nucleofection is a superb delivery technology for all these genome editing technologies. Nucleofection can be used to create sophisticated and elaborate diseased model systems in the following fashion. You can take cells from either a patient suffering from a genetic disease or a normal individual and then create iPSCs from these cells. You can then differentiate these terminally into the desired cell type. At the same time, you can take a portion of these cells and then modify them using CRISPR or one of the other technologies and to create an identical cell type, isogenic except for the CRISPR modification. These cells can then be differentiated in parallel with the normal cells, and then you can create a system where you have isogenic populations of cells, identical except for the CRISPR modification. It's human cells in a dish. You can also do this with cell lines. Many drug discovery groups have cell lines they've been working with for many years that are well characterized and well understood. You can now take these cells, CRISPR and modification into them, and now you'll have a pair of cells, cell lines that are identical except for the CRISPR modification. So now you can perhaps knock out a pathway and see how this changes the phenotype of the way this works in your assay. So it allows a sophisticated breakdown of what is going on in these cells. It allows you to manipulate things in ways that were difficult to do before, and particularly difficult to do in primary cells. This slide describes the formation of an elaborate genetic disease state model. This paper came from James Thompson's lab at the University of Madison, Wisconsin, and they used nucleofection to generate iPS cells using episomal-based plasmids. They differentiated and characterized these cells, and I won't get into the details except for the differentiation into hematopoietic progenitor cells by co-culturing these cells on OP9, a cell line which drives differentiation in that direction. They also made iPSC cells from patients suffering from chronic myeloid leukemia. These patients suffer from a translocation, the BCR-ABL translocation, also known as the Philadelphia chromosome, and is a unique characteristic of this disease. When they made iPSC cells for patients, the genetic integrity was maintained. So this BCR-ABL translocation was maintained in the cells, and this lays the foundation for the establishment of a disease state model of genetic diseases, because the iPSCs will keep the genetic architecture of that particular genetic disease. Here's an example of how easy it is to create iPS cells using nucleofection. Here we have CD34 cells that were grown in culture, transfected with the appropriate reprogramming plasmids using our 4D nucleofector. And you can see that by day 5 and 6, iPSC colonies begin to appear. By days 13 and 14, the cells are beautiful, and you're able to start picking colonies. And then in the last panel, 15 passages later, you can see that we have well-established iPSC lines. Using nucleofection, this is a very simple and straightforward prospect. These iPSCs that you generate can then be differentiated in many different directions. Here we have an example of the differentiation of iPSCs into hepatocytes. You can see with OCT4 in the pluripotency on day 0, OCT4 is expressed, but it disappears quickly as these cells begin to differentiate. You'll see other markers that will come up and then go down, such as SOC17. And you'll see other markers, later markers, that will come up and stay up, including albumin, which is a marker for hepatocyte differentiation, which is, goes up and then stays up. So you can take your iPSCs and differentiate them in a variety of different directions. Neuronal differentiation is straightforward and easy. We have customers who've done cardiomyocyte differentiation, and another customer who's even made lung epithelial cells from their iPSCs and then put them into an air-liquid interface for, again, advanced and sophisticated analyses. So this gives you the flexibility to create a model system of human cells in a dish with or without a CRISPR modification, so you can do direct comparisons. So using nucleofection as a central transfection technology, it gives you flexibility to work with primary and human cells effectively. iPSC generation using nucleofection is straightforward and easy, and nucleofection also facilitates the transfection of CRISPR substrates into iPSCs for specific modifications. And again, this results in 2 isogenic populations of human cells that are identical except for this CRISPR modification. These cells, these populations of cells can then be directly differentiated into terminally differentiated cell types for use in screening or cell-based assays, or for use as an elaborate model system for sophisticated analyses. So again, the end result is the 2 isogenic populations of differentiated human cell types, human cells in a dish, identical except for that specific modification, and again, in a dish where you can now do advanced analyses with them. Let's look at our nucleofector platform in this next slide. We have our 4D nucleofector, which is the benchtop device. It allows us to do either strips or cuvettes for this purpose. If you decide you need greater throughput, you can upgrade to the 96-well shuttle, which plugs into the 4D nucleofector system as part of the modular system, which will give you that greater throughput. The 384-well device is designed for high throughput screening. It's a standalone device, and it is designed to be integrated into robotic systems. The point here is that all of these systems are going to use the same solutions, the same programs, even the same cell numbers if you use this 20 microliter transfection throughout, and you'll get the same performance throughout. So you can scale things up or down without having to worry about reoptimization or having different results. This flexibility also applies to the large-scale 4D nucleofector LV unit. This is part of our modular system. The 4D core unit drives this system as well, and allows the transfection of large cell numbers ranging from 10 million up to 1 billion cells and even more. These conditions from the 4D nucleofector translate precisely into the LV nucleofector, so you can do your optimizations at a lower throughput with the 4DX unit, and then when you need to scale things up, it'll transition seamlessly into the LV unit. The LV can also be driven by the 21 CFR11 compliant software, or logware software, which will restrict access to authorized users. It will create audit trails and protect the integrity of the data. So this is all the things that are needed when you're doing things in the clinic. And this has also been tested on all kinds of cells, including clinically relevant cells like human T cells and CD34 cells, as well as relevant cell lines for protein production like CHOs, HEC293s, and K562s. Here we see an example of the scalability using human T cells. We have two programs that we tested here, EO115, which is our lower transfection efficiency but higher viability program, and then FI115, which is our stronger program, which also gives reasonable viability in this case. Here we compared 100 microliter transfections from the 4D nucleofector with two different versions of transfections with the LV unit, the 1 mil cartridge and then the flow-through LV cartridge. And you can see with EO115 that we had about 50% transfection efficiency across the board, whether it was the 100 microliter cuvette, the 1 mil cartridge, or 10 mils through the flow-through cartridge with very reasonable viabilities. With the stronger FI115 program, you can see a better transfection efficiency, around 70%, and then the viability is also maintained well across the different samples as we go from 100 microliters to 1 mil to 10 mils. Here's an example of the scalability of the transfection in human T cells of the CRISPR RNP protein. This was done with Matt Porteous at Stanford, and you can see that he tested and compared the X unit, the 4DX unit, with the LV nucleofector. This was the first try that he had on this, so this is N=1, and you can see that the results are very comparable to each other. I also wish to point out that with the X unit, he used 300 micrograms per mil of the Cas9 protein, twice as much as he used with the LV unit at 150 micrograms per mil. And you can see that the performance is still very comparable, even though he used half as much Cas9 protein here. So again, this is data demonstrating, functional data, that shows that the RNP CRISPR complex can be transfected effectively into clinically relevant cell types such as human T cells. Here's another example of human T cell transfection, in this case with zinc finger mRNA. The human T cells were transfected in small scale with 100 microliter transfections in the 4DX unit, or large-scale transfections using the 1 ml cartridge for the LV unit. And they transfected either GFP mRNA or zinc finger mRNA. The zinc finger mRNA transfections were assayed by cell 1 assay and indel formation. And you can see that in the first experiment, there was indel formation, but not at quite the same level as you saw with the LV unit. The LV actually had much better indel formation in this experiment. So this just shows you can also use zinc finger mRNAs and other genome editing technologies, as well as CRISPR with the LV unit. So what does this mean for your research? Basically, nucleofection is a premier transfection technology for the non-viral transfection of DNA and other substrates into primary cells and physiologically relevant cell lines. This includes clinically relevant cell types like human T cells or CD34 cells, which are involved in the most innovative treatments that are being currently done. Nucleofection will allow you to use the most relevant cells in your work, especially primary cells. These express the right receptors and pathways. They respond appropriately to stimuli, and the results from using primary cells are far more likely to translate into in vivo model systems. Nucleofection excels at the delivery of DNA and other substrates and is an ideal companion technology for genome editing technologies like zinc finger, tau, and CRISPR. It can really help facilitate the generation of the cells that can be used in next generation cell-based assays. Finally, our 4D nucleofector LV unit can transfect large cell numbers, clinically relevant numbers of cells, ranging from 10 million up to 2 billion cells at once. So this really allows it to play a role in innovative new therapies such as CAR-T and other cell-based therapies. So thank you for your attention, and I'll be happy to take any questions at this point. Thank you.
Excellent. Thanks very much, Greg. I think the audience now has a much greater appreciation for the advantages of nucleofection when using various genome editing modalities. So it looks like we've got some really great questions already that have come in, but I'm encouraging the audience to keep them coming, since this will be your final chance to submit questions for our speakers. Simply type your question into the Q&A box of your screen and hit submit. All right, so it looks like we have a lot of really good questions, so let's begin the Q&A session. Give us a few moments to get everything squared away on our end, and we will begin the Q&A. All right, everyone. Before we begin the Q&A, I just want to point out that we have great resources that our sponsor, Lonza, has given to you guys. There's a couple PDFs located in the resource widget box at the bottom right-hand side of your screen. So feel free to take a look at them, download them. They're really some really useful information for everyone. All right, so let's start the Q&A. We'll get to as many questions as we can. Matt, the first question is for you. One of our audience members would like to know, why use AAV rather than electroporation to deliver the template?
Yeah, that's a great question. I'm sorry I missed explaining why in my talk. And also, excuse me, I've got a hoarse voice. I haven't been drinking whiskey and smoking cigars all night. So in cancer cell lines, when you deliver plasmid DNA or naked DNA, for reasons we don't understand, they don't inactivate the innate sensing mechanisms, the toll-like receptor sensing mechanisms. But in primary cells like hematopoietic stem cells and T cells that we focused on in this webinar, when you introduce naked DNA into these cells, they get recognized by the pathogen sensors and activate an interferon response, making both the cells themselves and their neighboring cells very sick. So the reason we have to use AAV is as a way of getting the donor DNA across the cytoplasm into the nucleus without being sensed. So if you could find a non-viral way to traverse that minefield and get the DNA into the nucleus, that would work as well.
All right, great. Thanks very much, Matt. And we'll take any of that extra whiskey and cigars you have left on hand to send them our way. Greg, next question is for you. How many T cells can you transfect at the same time using nucleofection?
Good question. With our protocol, we recommend 1 to 5 million cells in our 100 microliter cuvette. But if you read the protocol, you can actually push this up to 20 million cells per cuvette. If you extend this into the LV, this means in the 1 ml cartridge, you basically multiply by 10. So that'd be 20 million cells times 10, 200 million cells in the 1 ml cartridge. This hasn't been tested, but I suspect this will be the case in the LV. And then if we do the 20 ml flow-through cartridge, you multiply that by 20. So we are conceivably talking about 4 billion T cells per transfection with the LV unit and the 20 ml flow-through. We haven't tested that. I think we could cut that in half and do 2 billion. I wouldn't hesitate about that at all. But I think if we wanted to push the system, we might be able to do even better.
And I'll add on to that. I know the question was directed at Greg, but we've been fortunate to test the LV unit, and we haven't done T cells at 200 million cells per mil on the LV unit, but we did do K562 cells, a cancer cell line at 200 million cells per mil, and it worked just fine. So I would expect that it would work.
All right, thank you, gentlemen. Matt, another question for you. Looks to be a little bit related to what you guys are talking about. What is the highest HR efficiency reported in vivo?
Yeah, so I think the current world record or published world record goes to Dan Anderson's group at MIT. It's published this year in Nature Biotechnology, and they achieved 6% HR frequency in the liver, where they combined delivering the CRISPR-Cas9 nuclease through a lipid nanoparticle, and then they delivered the donor through an AAV. So hopefully that record will be broken every year as we get higher and higher, but I think currently the record is 6%.
All right, thank you, Matt. Greg, another question for you. One of our audience members asked, do you have any experience in knocking a transgene, big size of about 3 kDa B, using HD approach and RNP?
Well, you're going to have to realize that this will change based on the cell type. There will be differences based on this efficiency in different cell types. So that's the first thing. I think the larger the insert size you're trying to put into the genome, the lower the efficiency is going to be. So small ones will get in much more efficiently, and I would imagine either a linear or just a decreasing efficiency as your insert gets bigger.
All righty, thank you, Greg. Matt, another question for you. How many micrograms of Cas9 protein and in vitro transcribed RNA and RNP complex in 20 microliters of lawn solution in strips? The audience member asked, they say, I work in T cells and have faced this problem exceeding maximum volume requirements for nucleophage.
Right. So we've done a dose titration, and I'm going to give you the concentrations per mil. And what we found is that actually scales across the 20 microliter, 100 microliter, and 1 mil conditions. So it's really what you want to think about is concentration per mil, and then do the adjustment per the volume you're using. And so what we have found is that using 150 to 300 micrograms of Cas9 per mil is the right amount, or it gives optimal amount. If you use more, you don't really get much more bang for your buck. And if you use less, you do start seeing a decrease in your on-target editing frequency. We complex the protein with 2.5 to 5 moles of guide RNA per mole of Cas9 protein. So again, you just have to do your calculations based on moles. But that's basically the range that we found. And again, that's those have been validated across K562s, T cells, and CD34 cells. I will note that one of the things you can do when you order Cas9 protein from commercial sources is they sell both low concentration and high concentration formulations. So you might want to order the higher concentration formulation, which I think comes at maybe 5 micrograms per microliter.
All righty, thank you, Matt. Oh, sure, go ahead, Rick, if you want to add to that.
I was going to just jump in there real quick. The way nucleophexin works is when we hit the cells with a pulse, we open up pores in the cell membrane and the nuclear membrane, and we stabilize these pores with these cell type-specific solutions that we have. And then whatever transfection takes place is really a passive diffusion event that happens. So the substrate, whatever substrate is present, will just passively diffuse into the cell. This has ramifications when you scale things up because this being dependent on concentration, you have to maintain the same substrate concentration in your volumes as you increase and scale things up. So as long as you maintain that concentration, you should get equivalent transfection across the different scales.
All right, thank you, Greg, and stay with us because we have another question for you. What is the best method or protocol to evaluate off-target activity?
This is difficult. I don't think there's any easy answer to this sort of thing, but it will be relevant to, as people move forward into the clinical realm, to evaluate what kinds of off-target effects take place. A lot of times people will look at their guide RNAs and then go through scan the genome to see what other similar sites there are with 1 or 2 mismatches. This is kind of like shining the spotlight on a particular area, and you can see what's happening there, but you can't see what's happening where you're not shining that light. Some people, there are kits out there that will tag the ends so you can see how many ends are being generated in a particular cell type, and so you can do this. Deep sequencing is one way of looking at this. You can sequence in from those ends. But it's going to be really difficult to look at off-target effects that don't follow the usual rules of the guide RNA with a few mismatches. And so that's one thing that we're always going to have to be aware of, that there will be off-target effects. However, by choosing things like the RNP, you'll be able to minimize this because these proteins are labile. You'll have a burst of nuclease activity, and then it's going to go away. And what seems to be the consensus is that the longer these nucleases are active in a cell, the more chance they have to scour and scan the genome and find these imperfect binding sites and cut the off-target effects. So by minimizing the time that these nucleases are active, you will, in general, decrease the amount of off-target activity that you see.
And I'll jump in on this as well. This issue of identifying off-target mutations is a rapidly evolving and moving field with no clear standard yet. So I really think you need to think about what you're doing with the technology in terms of being worried about that. I will point out that when people have done whole genome sequencing on CRISPR modified clones, they find no evidence of off-target activity. What we find is there's off-target activity when you look at in populations of cells, but in general, most cells will not have an off-target effect. It's only in populations that you start seeing this. So you really have to put this in context, and as I said, the moving field between biased approaches, which Greg described, and unbiased approaches, which Greg described, and there's new assays coming out every year, or every month almost.
All right. Again, thank you, gentlemen. Matt, another question for you. What prevents the CRISPR-Cas9 RNP complex to recut genomic target sites? Does it just keep cutting until it's not recognized anymore? And how about the cell therapy?
No, it does, and actually it goes to, so that's why when we do homologous recombination in our donor template, when we introduce a change, we actually also have to introduce changes to prevent recutting. So I think what you get is exactly as the person asking the question is suggesting, that the nuclease will cut at the target site until it's no longer, until the site has been mutated and it no longer recognizes the site, or until the nuclease is no longer around. Going to Greg's earlier answer, which I agree with, that one of the ways of minimizing off-target activity is have the nuclease disappear quickly, and so therefore it's less likely to find these off-target sites, but then it's also less likely to recut a on-target site that hasn't been mutated. So definitely the nuclease will bind and cut, and as long as that site is around and the nuclease is around.
Okay, great. Thanks, Matt. Greg, another question to stick with the off-target theme. One of our audience members asked, to what extent is the use of the RNP Cas9, does it address off-target related biosafety issues?
Well, I think we've already kind of addressed that, but basically the RNP comes in, it is immediately active because it's already complexed with the guide RNA, and you'll see this burst of activity, and then because it's labile, it'll go away. With an RNA approach, the mRNA for Cas9 could be co-transfected with the guide RNA. It would be translated, and then it would find the guide RNA and then begin action. So it would be a little bit delayed, kinetically speaking, from the action or activity of the RNP. If you look at DNA, put in a DNA expression plasmid, it's transient transfection, so these are around for 3 to 5, 7 days, cranking out Cas9 protein, cranking out the guide RNAs throughout this time. So there's a much greater opportunity for these complexes to scan the genome and cut at imperfect sites, leading to those off-target effects. So from a biosafety standpoint, I think the best we can do would be to limit the nuclease activity with the RNP, perhaps the RNA, and then that would be the best way to minimize off-target effects, and I guess that's your biosafety issue.
All right, thank you, Greg. And it looks like we have time for one more question for Matt. Matt, one of our audience members asks, when do you think genome editing techniques will be used to treat human monogenic genetic diseases like beta thalassemia?
Yeah, lots of people are excited about that. I think what we'll see is in either in the last part of this year or certainly in 2017, we're going to see CRISPR-mediated editing in T cells reach the clinic, and I would expect that we'll see CRISPR-mediated editing of hematopoietic stem cells or progenitor cells reach the clinic in 2018 for diseases such as sickle cell disease, beta thalassemia, severe combined immunodeficiency, and others. So I would expect to see those in 2018, and then for them to rapidly accelerate after that.
All right, thank you. Great,
Thank you.
Matt, for that one. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our site for up to a year at www.genengnews.com. So if you missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues. I'd like to thank Matt and Greg again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Lonza for sponsoring our. And don't forget to check out the resource widgets at the bottom of the screen there. Feel free to download any of those PDFs that are in there. So hopefully we'll see you again in another Gen webinar in the near future. Goodbye for now.
Thank you.
Okay, thank you.